A frameshift mutation in a patient with Tay Sach's disease causes premature termination and defective intracellular transport of a subunit of beta-hexosaminidase by Michelle M. H. Lau and Elizabeth F. Neufels. A little bit of background of Tay Sachs is that it is a disease that arises from a mutation on a gene called the hexagene. The hexagene yields subunit of beta-hexosaminidase. The enzyme consists of two different subunits, both the alpha and the beta, making it a heterodimer. Without both of these functional units, the enzyme itself is not functional. However, without functional beta-hexosaminidase, GM2 lipid cannot be broken down. This leads to its accumulation in the brain, causing cognitive impairment, such as seizures, among other neurodegenerative symptoms. There are several mutations that can ultimately lead to the disease of Tay Sachs. However, the molecular mechanisms that are involved in each of these are what is different between the different mutations, causing the defective beta-hexosaminidase to be produced.
So some mutations of beta-hexosaminidase, such as the insertion that occurs in exon 11, which is the most prevalent mutation which occurs in Eskenazi Jews, leads to a profound deficiency of mature mRNA as well as a deficiency of polypeptide production, which in this case is the hexosaminidase subunit. And furthermore, since there is little to no functional hexosaminidase, GM2 cannot be broken down, so it accumulates rather quickly in the brain, which leads to the typical onset of Tay Sachs, which occurs in very young children. However, there are other mutations which are much less severe and allow for the expression of some functional hexosaminidase. Furthermore, there is some breakdown, but still not enough to prevent accumulation of GM2. It just accumulates more slowly than it would in typical Tay Sachs. So this is labeled as late onset Tay Sachs, which is the Tay Sachs that's observed in the patient that's in this study.
In this study, it was observed that in an Italian patient, truncated alpha subunits that were produced were inserted into the lumen of the ER and failed to undergo post-translational processing. Typically, if this happens and it fails to undergo post-translational modification, it leads for it to be secreted. However, in this individual, it was observed that the mutant alpha subunit was trapped instead in a compartment of the ER and was later degraded.
The two questions that this paper is trying to answer is they are trying to determine whether the smaller peptide size was linked to post-translational modification. They were also trying to find out whether what the mutation that causes the truncated beta-hexosaminidase was, and why it led to the subsequent phenotype.
In order to determine whether truncated protein size was linked to post-translational modifications, the methods that were carried out for this was that proteins were first synthesized in cell-free environments. This was done by eliminating cellular components, therefore inhibiting post-translational modifications. RNA was then derived from the patient's cells, the patient's father cells, and from normal fibroblasts. RNA was translated in the presence of rabbit reticulocyte lysate, which contains necessary components for protein synthesis. This is helpful because when eliminating cellular components, nutrients are still available for protein synthesis to still take place. 35S methionine was a tool that allowed for good visualization of protein synthesis that was taking place. Proteins were then isolated by immunoprecipitation and electrophoresis. Immunoprecipitation is a procedure that involves the addition of antibodies in order for coagulation to take place and for the proper separation or isolation of the proteins that were targeted. The second big question was to find what mutation causes truncated beta hexosaminidase A. The methods that were done to answer this question was the amino and carboxyl terminants and were radio labeled in order to help with visualization. Immunoprecipitation was carried out as well, but was performed to test for the presence of carboxyl and amino sequences. Reverse transcriptase of mRNA and PCR were also carried out, followed by direct sequencing. PCR and direct sequencing of DNA was performed, and DNA fragments were hybridized to allele-specific oligonucleotide probes. For confirmation that in fact the mutation did cause truncation of the proteins, a plasmid was created and expressed into the CO1 cells. This then showed that the same phenotype as the patient was observed, where the protein is retained into the ER other than being secreted. In the results, this image shows the electrophoretic mobility of the alpha subunits. IMR represents the normal alpha subunit, 1051 represents the patient that has Tay Sach, 1054 is the parent of the patient. It can be seen that 1051 and 1054 traveled further, suggesting that they are smaller in size in comparison to IMR 90. However, when thinking back to the big question about whether protein size is linked to post-translational modification, the answer is then no. This is due to the fact that even though 1051 and 1054 were synthesized in a cell-free environment, they still were truncated, which can be explained by the fact that they are smaller in size, allowing them to travel further in the gel.
So, when observing the previous gel electrophoresis, they also determined that because of the distance difference, there was approximately a 2 to 3 kilodalton difference in molecular mass, which they believe could be accounted for by approximately a 20 to 30 amino acid difference. And these amino acids were present in the original, but not present in the 1051. This also raised some questions when they did the immunoprecipitation with that specifically targeted the carboxyl and the amino ends. When they did the immunoprecipitation that targeted the amino ends, they observed that the 1051 and the IMR90 precipitation was equal if they removed the signal sequence that they used to visualize the 1051. However, when they compared the carboxyl ends, they determined that there was no immunoprecipitation in the carboxyl regions for the 1051, and they believe that this could be explained by the fact that there was no amino acids present in this region for the antibodies to target. Furthermore, there was no precipitation occurring. This led them to focus in on this area, and so that's when they conducted the reverse transcription and the PCR of this area by the 3' end. And then they directly sequenced, which can be observed in image A. When they directly sequenced, they determined that the 1051 had a deficit of a cytosine, which can be observed in the rightmost lane in the 1051. That was typically present in normal individuals in the IM90. Furthermore, this indicates a cytosine deletion. When they took the sequence information and took it over to a translation where they observed the amino acids, on the top you can see a normal individual sequence where the C is present. Typically after the phenylalanine there's an arginine. However, after the deletion, that arginine is replaced by an alanine, and somewhere downstream there is a termination codon, which subsequently leads to the truncated protein, which was observed in the Italian individual. Furthermore, this gives insight into what the mutation that causes truncation and the remainder of the phenotype is. In image B, is what they call a dot blot analysis, and ultimately 1053 and 1054 are the individual's parents, and this just shows that they were heterozygotes. It also shows that in 1051 the individual is a homozygote for this deletion. Going back to the previously mentioned of the COS transfection, it just showed as a confirmation to outline the relationship between the deletion and subsequently the phenotype expression.
In this study, the reason for why the mutated protein is retained is currently unknown. Therefore, for future research, looking into the reason for why the retention of the WG1051 alpha unit was retained in the ER would be a potential start for working towards a treatment beneficial to Tay Sach's patients that suffer from this mutation. By understanding why the protein is retained, interference could then be made in order to halt degradation and allow some partially functional hexosaminidase to be produced. This would then prevent GM2 accumulation and would prevent the arise of the phenotypes that are exhibited.